Hello and welcome. This is Morgan Abeka Isotox at Kamajun. For today, I'm going to present about metabolism in biochemistry. Okay, so to start with, we'll start by defining what is metabolism. So, metabolism simply involves something like these are the biochemical reactions that occurs within our body. So as you can see on the diagram here, we've got the fuel, the one that powers up energy in form of ATP, that is the adenosine triphosphate. Then energies are made in the liver. Yeah, and then we've got the biochemical reaction under biochemistry and how all these things works within the metabolic reaction. Okay, so without wasting much of your time, we'll look at the objectives that we have today. So the first objective that we have is we are going to define what metabolism is, and the second objective, we are going to understand, like, why is it very important for us to understand the metabolic pathways. So it's very much important for us to understand these metabolic pathways. Then the third objectives, we are going to assess the normal metabolism versus the abnormal metabolism. Yeah, because you can't know what is abnormal if you don't know what is normal. So it is only when you understand the normal metabolism that's when you can be able to ascertain any abnormalities that will arise thereafter. In certain situations, medical situations, or rather clinical situations such as malnutrition or marasmus. Then the fourth one, we are going to look at how metabolism is regulated. Okay, so let's look at what is metabolism. Okay, metabolism. What is metabolism? So metabolism is just the sum of chemical reaction that takes place, or rather that occurs within its cell of a living organism, and that provides energy for vital process and for the synthesizing of organic material. Okay, so what you need to understand is metabolism. These are the chemical reaction that takes place within the cells. So now when you look at what a cell is just the basic structure, living thing of it's the basic structure, uh, it's the basic structure living thing of it's a basic structure of a living organism. That's what a cell is. So within a cell, there are these chemical reaction that occurs. To be specific, these biochemical reaction that occurs, they occur within the cytosol. That is, under the cytosol, we tend to see glycolysis, and within the mitochondria, that is, we tend to see the Krebs cycle or the tricarboxylic acid cycle, of which it involves the synthesis as well as the breaking down of these chemical reactions. Okay, so let's so after understanding the definition for metabolism, I'll now introduce you to the metabolic pathways. So initially, there are three major metabolic pathways, which include anabolic pathway, catabolic pathway, and antibiotic pathway. So anabolic pathway, it is the pathway which synthesizes the monomers. Now you need to understand the word monomer there refers to small molecules in form of, it can be glucose, to when you are using glucose to synthesize carbohydrate, or maybe when you're using the three fatty acids plus glycerol to give you lipids, or maybe when you're using amino acids to get proteins. Yeah, so it involves the use of monomers, which are small molecules, to make polymers. Polymers, these are large molecules such as proteins, lipids, and carbohydrates. And it is endergonic. So when we say endergonic reaction, meaning that the kind of the reaction that usually takes energy from the surrounding cells, or from the surrounding environment. Okay. Then the second one is and then scenarios where we can see an anabolic pathway is when someone is when someone is feasting. You realize, you say that metabolic disorders that will come, All right. Okay, let's continue. The metabolic disorders that will come during anabolic pathway, anabolic disorders, they might involve like obesity. Yeah, those ones, yes. Then other ones, like for example, let's look at catabolic pathway. So under catabolic pathway, this is the pathway that involves the breaking down. Okay, so it has to be the breaking down of polymers to form monomers. So you're breaking down large molecules to form smaller molecules. And it is hexagonic reaction. That simply means that it releases energy to the surrounding. Okay, so this one, it releases energy to the surrounding cells. And this one, catabolic, the normal physiological way that we can see catabolic pathway is when someone is fasting or starving. Those are the pathways, those are the only examples that we can appreciate catabolic pathway. Then amphibolic pathway, this one, it acts as a linkage. It links anabolic pathway as well as catabolic pathway. This one, it acts like an inhibitor, the one that it inhibits and activates. So meaning that when one process is on, like for example, if catabolic pathway is on, is activated, then the anabolic pathway will be inhibited. Or maybe if anabolic pathway is activated, then catabolic pathway will be inhibited. Okay, so now let's look at the summary of metabolic pathway. So in the summary of metabolic pathway, what do we see? For example, if you ingest proteins, proteins will be broken down into amino acids, and these amino acids, they are going to form amino acid pool, of which from after the protein synthesis, where amino acids have been broken down under the chemical reaction that occurs within the cell, we tend to produce urea. Now, all these things, they occur within the liver. So it is from where, because the liver is the one that usually do deamination, where protein synthesis and amino acids, the amino acid pool is now being converted and degraded into urea, and then other protein goes into the tissue. Then it also, from there, we tend to, these amino acids that are broken into the pool, they usually form what we call pyruvate. So once they form pyruvate, and then this pyruvate is the one now which the pyruvate is the one that is insoluble to the mitochondria matrix. It can't enter the mitochondria matrix, so it has to be converted to acetyl-CoA. Okay. Again, these proteins, will usually find them in the Krebs cycle. Like for example, we've got the succinyl, the succinate, and those other ketoglutarate, those are other proteins that we get to find in the citrate. Those we get to find them in the Krebs cycle. Okay. Now, what happens if someone has taken carbohydrates? When you take carbohydrates, they'll be broken down to monosaccharide, which are just glucose. So monosaccharide, these are simple glucose molecules, of which we get the glucose. Now, the conversion of glucose to glycogen, it's called glucogenesis. It's called glycogenesis. So under glycogenesis, this is where all glucose are converted into glycogen by the hormone called insulin, the one that is produced by the beta cells the beta cells of the islet of Langerhans that is found in the pancreas. Okay. Then the conversion of glycogen to glucose, that one is done, it is covered through the process that is called glycolysis. So we call it glycolysis. So glycogenesis, the formation of glycogen using glucose. Glycolysis is the breaking down of glycogen to form glucose. Now, when you look at these two, there are two hormones that are produced by the pancreas. Inside the pancreas, there is a certain component that we call the islet of Langerhans, the one that produces two cells. It produces the alpha cell and the beta cell. So the alpha cell, it is only active it's only active to low blood glucose level, and it's the one that usually produces an hormone called glucagon hormone. And then the beta cells, the B cells, these beta cells, what they do is that they are sensitive to hyperglycemia, that is an elevated level of glucose in blood. So they usually release an insulin hormone, the one that usually works on the excess glucose and convert them into glycogen by the process known as glycogenesis. Okay, so when one process is working, when glycogenesis is active, meaning glycolysis has to be inhibited. So that's how it works. Then how about if someone has ingested lipids, which are the triglycerides? So if someone has ingested triglyceride, these triglycerides are going to be broken down to form glycerol, that is glycerol and the 3 fatty acids. So these are the precursors of the lipids. The precursors for lipids, these are the 3 fatty acids and glycerol. And then these 3 fatty acids and glycerol, they can be converted and form pyruvate by glycolysis. Okay. When that is done, again, these 3 fatty acids, the 3 fatty acids as well as the glycerol, they can also form triglycerol. They can also form triglycerol. And then the triglyceride can now form what we call acetyl-CoA by the process called fatty acid oxidation. Now, this one you are going to understand it when we start looking at lipid metabolism, because this is where we have the beta oxidation, the fatty acid oxidation, the beta oxidation, on how these fats are being oxidized, like they are being oxidized to form acetyl-CoA. Okay, so now when pyruvate has been formed, you need to understand that pyruvate is an insoluble molecule that cannot enter the matrix of the mitochondria. Hence, it needs an enzyme called pyruvate dehydrogenase, which is a triose enzyme. This enzyme is the only enzyme that can convert pyruvate to form acetyl-CoA. When acetyl-CoA has been formed, this is the only molecule that has got the triose carbon, the 3 carbon molecules. And it's the only molecule that is soluble, that can enter and penetrate the matrix of the mitochondria to activate the Krebs cycle. When it enters in the Krebs cycle, which is the tricarboxylic acid cycle, what is it going to do? It is going to find oxaloacetate seated waiting. And the moment it finds oxaloacetate there, oxaloacetate, it is going to initiate the conversion of oxaloacetate to citrate. And then the enzyme that ensures that this conversion is done, it is called citrate synthase. And when citrate has been formed, the very citrate will be converted and form cis-citrate by the enzyme called aconitas. And then when the cis-citrate has been formed, the cis-citrate will be converted to form isocitrate by the same enzyme called aconitas. And then when the isocitrate has been formed, this isocitrate will be converted to form what we call alpha-ketoglutarate by the enzyme called isocitrate dehydrogenase. And when this alpha-ketoglutarate has been formed, this alpha-ketoglutarate, the one that has been formed, it is also going to be converted to form succinyl-CoA. When it forms succinyl-CoA, then the enzyme that does this conversion is called alpha-ketoglutarate dehydrogenase. The succinyl-CoA, it also going to be converted to form succinate by the enzyme called succinyl-CoA thiokinase. And then when the succinyl-CoA has been converted, this succinyl-CoA is also going to form what we call, fumarate. Then from fumarate, the enzyme that does that conversion from converting succinate to fumarate, we usually call it as in succinate dehydrogenase. Then from there, we tend to have fumarate. Fumarate forms marate. Now, the formation of marate from fumarate, there is an enzyme that does that conversion that we call fumarase. It is called fumarase. And then the conversion of malate to oxaloacetate. It is done by an enzyme that we call malate dehydrogenase. And then when oxaloacetate has been formed, it will also wait for another acetyl CoA molecule for this Krebs cycle wheel to spin again. And this is how the mitochondria powers the cell with energy. It is the pump that generates ATP, the adenosine triphosphate, by the use of nicotinamide diphosphate. All right, okay. So this is just the summary of the metabolic pathways that occurs within. Then from the Krebs cycle, it goes to what we call the electron transport chain. This is where now hydrogen is used and electrons are used to produce other form of what? ATP. Okay. Now, what is normal metabolism? Why is it very much important for you to understand the knowledge of normal metabolism? The knowledge of normal metabolism, it helps to ascertain metabolic abnormalities that will arise thereafter. If you can't identify what is normal, then you can't tell what is abnormal. It is by virtue of you understanding what is normal, then that will give you the capability or the capacity, the synesis or the phronesis, the acuity and the profundity to understand any abnormal thing that will arise thereafter. So, the normal or rather the abnormal metabolism, these are just disorders that arise due to malnutrition or rather obesity. For example, if someone has been on starvation for some time, they will tend to have what we call catabolic abnormalities or catabolic disorders, where the body will use up all its energy, and then these people will be malnourished. So that one is called malnutrition. And a person can become obese in an event where this person has been feasting too much, eating too much, abnormal appetite. It can make someone to be obese. All right. So these are what we call abnormal metabolics. Okay. Then the normal physiological metabolism. This one, it includes when someone is fasting, or rather when someone is on starvation. That's just normal. Okay. All right. That was just a commercial break. Yeah, it was okay. Let's continue now. So, we'll look at the biomedical importance of metabolism. Why is it that metabolism is very important? What are some of the importance of metabolism? Biomedical importance of metabolism. Number one, it provides a sound understanding of many diseases, which is very true. Once you understand what metabolism is, it gives you that inner understanding of many diseases that comes due to some metabolic abnormalities. And also it helps us to understand the variation and adaptation due to starvation. Because there is variation. These are certain changes that will change when someone is on starvation. And adaptation. These are certain things that will change. Like for example, if someone is on starvation, they tend to adapt to the same starvation. And these include exercise, pregnancy, and lactation. These are included in the normal metabolism. So, in normal metabolism, if someone is exercising, someone is pregnant, someone who is on lactation, these are the only thing that these we call them as in the physiological normal metabolism. These are under physiology. They are under the normal metabolism. Then the abnormal metabolism. These results from nutritional deficiency. So they result from either nutritional deficiency or nutrition deficiency, which includes like someone is lacking certain body dietary or maybe certain nutrients that the body needs. Then also enzyme deficiency. So on enzyme deficiency, this is where you lack certain enzyme to convert certain metabolic reaction. And also excess secretion of hormone. You realize that when there is an excess secretion of hormone, people tend to have, like for example, give you an example. If there is an excess secretion of the thyroid hormone, Someone will have hyperthyroidism, which is giantism. So those things will start occurring. So that one, it is as a result of what? Excessive secretion of hormone. Then also diabetes mellitus. So diabetes mellitus, this is just an example of a disease that is caused by abnormal metabolism. So abnormal metabolism can lead to diabetes mellitus. So it's very much important for you to understand the biomedical importance of what? Metabolism, because this will give you a vast understanding. Yeah. Then how is metabolism regulated? So regulation of metabolism, this one we can say, how is metabolism regulated? Number one, we need to start with enzymatic regulation. We need to understand there are certain enzymes that need to regulate metabolism. Meaning that we need to have enzymes that can activate or inhibit. For example, if someone is on, if someone has, like for example, if there is an increase, if the body has noticed that there is a rise in the blood, there is an increase in the blood glucose level, that is hyperglycemia. So what will happen is that the B cells, they will notice, because these B cells, they are sensitive to high blood glucose level. As they are going to produce, or rather they are going to activate the release of an hormone called insulin to convert glycogen, to convert glucose into glycogen. That process is called glycogenesis. Then the process is called glycogenesis. Again, when the body notices to say that there is low blood glucose levels, that is hypoglycemia, you call it hypo-G, meaning that when you're feeling weak in the muscles and the muscles are even twitching, that one is hypo-G. So the moment there are certain cells which we call the A cells, the alpha cells. These alpha cells that are found in the pancreas, the eyesight of Langerhans, these ones, they're very much sensitive to notice the low glucose levels, which is hypoglycemia. And once they sense that there is low glucose levels, what they will do is that they will release an hormone called glucagon. And once glucagon hormone has been released, it is going to break down glycogen to glucose. And that process is called glycolysis. It's called glycolysis. Okay. Then, so that is how enzymes help to regulate metabolism. So, when one enzyme is active, the other enzyme has to be inhibited. The other, in simpler terms, the word inhibit simply means to stop or prevent it. Alright, okay. Then other pathway regulator, we've got the negative feedback as well as positive feedback. So the positive feedback and the negative feedback, this one, when one is active, the other one has to be inactive. So this is, even these negative feedback and positive feedback, they also help to regulate the normal one, metabolism. Now, if these regulators, they are not regulating, they are not being activated or being inhibited, meaning that that's now a metabolic disorder. If hormones cannot be stopped, they're still coming, they're still being secreted, then that's a metabolic disorder. That is, you need to know that it's a metabolic disorder. Then also isoenzyme. These are two or more proteins that catalyze the same reaction, but they've got different kinetics. Like for example, the regulator properties, as well as what? The cellular distribution, and which are coded by different genes or by alternative what? Splicing. Okay. Then we also have what we call the reversible covariant modification. So under the reversible covariant modification, this is where we've got. Have you ever heard of the reaction that are reversible? That can be reversed by either some enzyme or an enzyme that is same enzyme or another enzyme. So such kind of reaction that can be reversed, also they can be regulated as well. Meaning that when one process is active, the other process has to be inactive. Then we also have what we call the compartmentalization. So under this is where we've got the multi-enzyme complex, where you find to say that and multifunctional enzymes. These are metabolites channeling. They will channel certain metabolite against to another channel, where these channels, the channeling of reactants and the channeling of reactants between active sites, like enzymes, they've got what we call the active sites as well as the active site where the enzyme might bind. So all those activation, they need to be done via what we call the metabolites. Metabolites, they need to be done by what we call metabolites channeling. Okay. Then there is also what we call transcription. Now, this one is done under nucleic acid. So you really need to know that under nucleic acid, we've got we have DNA and RNA. DNA, that is the deoxyribonucleic acid, which has got the sugar called deoxyribose sugar, and this one is the one that when it undergo replication, what we call DNA replication, usually form RNA by the process known as transcription. So transcription is the process by which RNA is formed from DNA. Then we have translation. So under translation, so when DNA forms, remember what you need to understand, that a cell is just the basic structural function unit of a living organism. And this cell, it has got the cytoplasm. And that cytoplasm, it has the ability to divide. When the cytoplasm divides, that process, we call it cytokinesis. That is the division of the cytoplasm. And then we also have the nucleus. Remember, the nucleus, it monitors all the activities that are occurring within the cell. Not only that, it also stores the genetic information that coded inside that is coded or rather embedded within the nucleus. We usually call it DNA. Okay, so even the nucleus can divide by the process that we call cardiokinesis. So inside the nucleus there is the DNA, and inside the DNA we have the chromosomes, which are the thread-like structure that codes for genes. And the gene is just a sequence of nitrogenous bases that codes for a particular protein. Yeah, so what you need to know is that when translation occur, like when transcription occur, DNA is going to form RNA, which is the ribonucleic acid. Now, this RNA that is formed, it comes in three types. We have the messenger RNA, which is the RNA that's used to send messages from other organelles. Like it's the one that carries message from the DNA. It's the one that transfers energy from the DNA to the newly synthesized protein, which are synthesized by what we call the rRNA, which is the ribosomal RNA. So the ribosomal RNA, which is the second RNA that is formed by transcription, this one, what it does is that it manufactures new proteins. Then we've got the tRNA, which is the transfer RNA. So the function of the transfer RNA, what it does is that this one, it transfers all the codings from the DNA to the newly synthesized protein so that they can appear same with the DNA, so that replication can occur. And these proteins are degraded. So protein degradation, this is where now we find to say that all the codings, all the information that was embedded inside the DNA are now being transferred into the newly synthesized proteins. Alright, thank you for your attention. This has been Morgan Epika Isodoxa Kama Junior Basanko Abamasute Basanko Abamandevu Apamonze. Okay, so if you're new to my channel, you can subscribe. Yeah, this is my YouTube channel, and then so I'm yet to publish another episode in biochemistry on bioenergetics, glycogenesis, and then Krebs cycle, and so many more others to come until we reach up to the heme production, the heme metabolism. Alright, okay. Thank you for your time. If you've got question or anything, you can comment in the box below, and then you can use the link that I've just attached or rather pinned to the video below. Alright, this has been Morgan Okay, so dogs, shalom.